Hello, my name is Joshua Ferrar, and I'm a junior biochemistry major. I'm an undergraduate researcher in Dr. Choi's lab in the biochemistry department on the health sciences campus. And my project is on the conformational changes of syntaxin 3B in regulating SNARE complex assembly. So a little background on syntaxin 3B is syntaxin 3B regulates SNARE assemblies in the retina, which is in the back of the eye here. SNARE assembly happens when light is sensed by the photoreceptors in the retina and converted to electrical signals that are processed at the active zone that is highlighted in the synapse picture in the middle here. And this is followed by synaptic vesicle tethering to the ribbon by the SNARE complex consisting of syntaxin 3B, synaptobrevin, and SNAP25, which can be seen in this last picture here. So what we hypothesized from this was that syntaxin 3B, the wild type, is predominantly in a closed conformation, not allowing this to happen. But a phosphorylation on the residue 14 of syntaxin 3B promotes it to have an open conformation and allowing this to happen. And how we did that was, so we started out by growing syntaxin 3B in the phosphomimetic mutant in an antibiotic solution. When the growth was finished, the proteins were then purified using the nickel affinity through wash processes that finish with the final elution using imidazole. The histine DAG in the salt concentration is changed using dialysis overnight and in a low salt buffer, so the next day the sample can be run through the high trap Q with increasing salt concentration. High trap Q will further isolate the protein based on the charge and fractions that are pulled and run on the gel the next day to confirm the protein is there. During growth, the ABI tag is biotinylated and used to immobilize the protein on the microscope slide. The protein is then labeled overnight with Alexa 555 and 647 and run through the G50 column, as shown to separate the free dye. As you can see here, you have the free dye up top and the labeled protein down on the bottom. All of this leads up to the use of cutting-edge technology of single molecule fluorescent resonance energy transfer, or SMFRET for short. And this technique allows us to analyze the conformation of a single molecule by exciting the molecules on the slide with light from the lasers. In this two-laser system, we excite the donor dye, and the emission will then excite the acceptor dye depending on the distance. We then determine the distance between the donor and the acceptor, resulting in either high FRET or close interactions, which means it is in a closed conformation, so there's little distance between the donor and the acceptor, which is what is shown in the figure up here. Yeah, or low FRET or far interactions, which means it's in an open conformation, so there is some distance between the donor and the acceptor as shown here. For our experiments, we expect to see low FRET because we expect syntaxin 3B to be in an open conformation, because in a closed conformation will not function properly, as I stated before. And we saw that syntaxin 3B was predominantly in the closed conformation, and which can be seen on this graph here, which can be seen on this bar graph, sorry, where there's a low percent of the population is in an open conformation. So we saw a lot of high FRET. But for the phosphomimetic mutant, we saw more low FRET, so we saw a lot more open population, as can be seen here. And this open conformation allows us to then see that it will function properly when creating the ternary SNARE complexes, as can be seen back here. And using this information, what we want to look at in the future is we want to introduce SNAP25 and synaptobrevin, because they are also a part of the ternary SNARE complex, as can be seen in this picture. And as well as we want to look at the conformational changes that will happen when MUNC18 is introduced, because usually syntaxin has a closed conformation as well with that. And that is my presentation. I would like to thank Dr. Choi and Claire for allowing me to work on this project. And I would like to thank all of you for listening to my presentation. Thank you.